Today, I want to give you an overview, a perspective of how we look at the origins of RNA and DNA and what we are primarily interested in. So we mainly use methods of quantum chemistry, and our main motivation here have been the challenges in the prebiotic formation of those two key informational polymers on the early Earth. So when we start thinking about how the building blocks could have been formed, there are many challenges associated with this. And one of them is simply the formation of nucleotides. And we tend to think about the formation of nucleotides as we are taught in high school. So we use this modular approach where we just think about connecting ribose to nucleobases, but this apparently doesn't really work, and the reasons are simply thermodynamic and kinetic. So there is a high barrier, but also thermodynamically you really go, the reaction goes with positive delta G, positive Gibbs free energy change. So it doesn't work in aqueous environment. It could work a little bit in wet-dry cycles, but even if you somehow bypass or resolve slightly this problem, then you still have trouble with really the stereoselectivity. So you could form many different pentose sugars with different orientations of those hydroxyl groups here. And the question is how you actually can select for ribose prebiotically, and this is really a huge challenge. So we are trying to show possible ways of resolving that. Basically, we have some interesting ideas how to do it. But even if you form RNA nucleotides, then you end up with another problem, which is RNA polymerization. But of course, you don't want to polymerize RNA randomly. So really, the key question is how RNA could have started copying itself, and how you create meaningful functional RNAs that could do something on the early Earth, maybe in conjunction with some simple peptides. So these are all of our motivations and the challenges that we are trying to address. And I'll show to you how we actually do it from the point of view of quantum chemistry and computational chemistry. So one of the key questions that we ask is what environmental factors favor the formation of RNA, because of course you could ask the question, okay, maybe there could have been something else, something different than RNA, a proto-informational polymer. We think that not. We think that RNA is very unique, and one of its remarkable properties is resistance to UV light. And this is what you will see here. So we find this part really fascinating, mainly because sun was slightly more active in the ultraviolet spectral range, but also there was no oxygen and ozone on the early Earth. I mean, molecular oxygen, which could have shielded the surface from UV light. So UV light could have been an important source of energy for prebiotic reactions and a selection factor for prebiotic molecules. And there were hypotheses which suggested that maybe biomolecular building blocks like the nucleobases were selected based on their photostability. So we contributed a little bit to this story by showing that many alternative nucleobases, which look slightly different to those that we know very well, so they have different substituents or different configuration of those substituents, they are actually not photostable, and the ones that are biological, they are very photostable. But there is another important aspect that we show, and this is really related to the formation of RNA and DNA building blocks. And here we have really a sequence of interesting contributions through which we show that UV light actually allows for very selective chemical transformations, very selective chemistry. So when you irradiate the mixture, you can actually promote reactions which otherwise wouldn't work. Reactions leading to the biological forms of nucleosides. So here you can see the pyrimidine nucleosides is summarized in this work of ours and our collaborators. But also it purifies the mixture of products. And what we had until very recently was this, which was the synthesis of the pyrimidine building blocks of RNA. But what we also recently showed in collaboration with our co-workers from Cambridge and from UCL London was the path leading to the purine building blocks. So they actually have some more works which also use UV, and we contributed to one of them. So what you can see here is actually quite a lot of interesting reactions, and the key starting compound that I'm showing to you is this, which is aminooxazolene, or you can even look slightly earlier at this small ring, which is really fragment of a sugar and fragment of a nucleobase. So basically what you have to do to bypass this problem of glycosylation, which I showed very early on the first slide, is you form an intermediate which has a preformed glycosidic bond. And when you form this intermediate, you can essentially access all possible nucleoside building blocks, and you can use the chemistry of small prebiotic molecules like HCN, urea, thiourea, HCN homologs, essentially, which allows you to form also the purine precursors. So you can form both the pyrimidine and the purine precursors. And these purine precursors can actually be selectively transformed into DNA purine nucleosides. And what our colleagues showed a year or two later, after this work, you can also form the RNA forms of those. You basically can do it through UV irradiation. And what we show is this remarkable selectivity and explain this selectivity based on quantum chemical simulations. We also try to predict some of the photochemical reactivity, which they might not show. So here we have a photoreduction chemistry, which really removes the sulfur after the absorption of UV photons. And as the reductant, you can use either bisulfite or you can use hydrogen sulfides. What is very interesting is the difference in selectivity. So hydrogen sulfide is not so selective. Bisulfite gives you very selective photochemistry. But in the end, what we are right now showing with one of my students is this reaction, or the irradiation of these products here. So the biological and non-biological forms of adenosine also yields only the biological one. So this one is not photostable, this one is photostable. So these are our early results. So what we actually do in terms of quantum chemical calculations is we try to explain reaction paths, and we really look at landscapes. Oh, okay, I wanted to go here. And we look at landscapes of energy. So we look at the potential energy and free energy landscapes. And when you think about reactions and even simple textbooks at the level of, well, maybe a little bit beyond high school, right? Then you always look at reactions as going through a barrier, right? So a very nice analogy is hiking. And what really matters is the structure of the molecule on top of that barrier and the energy, the barrier height, which tells you about the kinetics. The structure tells you about the mechanism. Now, when you think about photochemistry, really you can use a very similar analogy. It's just slightly different. Because you simply add some kick of energy into the molecule. And this excess energy you can simply imagine in terms of a skiing lift taking the skier to the top of the mountain. And this is essentially what happens to the molecule as well. So it gets a lot of additional energy from photon absorption. And really what happens afterwards is the skier selects specific skiing routes. And what really is essential are the intersections of those skiing routes. They decide where the skier will end the journey. So now when you think about, or when you think that those skiing routes are electronic states, you can quickly realize that the intersections of electronic states are necessary to understand the mechanism of photochemical reactions. And when you know the geometry and the structure of the molecule and the energy of that intersection, you can actually predict reactivity, or most certainly you can actually explain specific reaction mechanisms. And this is what we do. So we look at those energy landscapes, we calculate them for specific molecules. And just to give you an example, for example, if you want to predict or understand whether a molecule is photostable or not, what you have to look for are those peaked state crossings or conical intersections, because they can be accessed in a barrierless manner by the molecule, whereas those which are sloped, they cannot be accessed in a barrierless manner. Actually, the skier would have to climb there. So now when you have a molecule, it needs at least several vibrational periods to get there. And during this time, it is in a very reactive electronic state, a biradical state, which can react with something from the environment, like with a solvent molecule or with another reactant molecule. That can be destructive photochemistry or productive photochemistry, of course. But this way, just by looking at those landscapes and what happens to the structures of those molecules, you can tell quite a lot. And this is really what we do. And an example is here. And this is work done by my former PhD student who now is really developing his very nice independent career. And he figured out that actually sulfides can transfer electrons very selectively. So this is done by Mikołaj Janicki, sorry, I haven't mentioned the name. And he showed that sulfides can transfer electrons very selectively upon absorption of a UV photon, and they sulfides interact with the heteroaromatic ring of our thione hydronucleosides, so our intermediate towards purine nucleosides. So this interaction is really called chalcogen bonding interaction, and that enables for the breaking of that bond and then subsequently for excited state sulfur loss. So this way we actually explained why sulfides are not selective, because that works for both of these thione hydro intermediates to purine nucleosides, whereas when bisulfite releases hydrated electrons to water, those hydrated electrons can actually do very different things depending on the actual intermediate that you have. So when you look at the biological precursor to nucleosides, you see the same type of photoreduction chemistry, whereas when the hydrated electron attaches to the non-biological precursor, what really happens is puckering of the ring that can be followed by breaking of that bond and some hydrolytic reactions which essentially decompose that compound. So it becomes much less photostable when you expose it to UV light in the presence of bisulfite. Okay, so this is part of the story which is related to the selective formation of nucleosides. And you could ask the question now, okay, so you use UV light to generate nucleosides, but we all know that RNA and DNA is susceptible to photodamage when exposed to UV light. So how do you solve that problem prebiotically? Because there were no enzymes. And some of the most frequent damages are thymine dimers. And those thymine dimers, they essentially change the structure of DNA. They block its biochemical activity and functions. And what nature does, it actually has those elaborate enzymes which can locate the place of the lesion. They can suck it into the active site and transfer the electron by breaking that cyclobutane, and break that cyclobutane ring afterwards, after the electron transfer. So these are very efficient enzymes. They are called photolyases. And we envisaged that maybe specific nucleobases could do the same, but maybe some non-canonical nucleobases that could still do some productive and interesting work. And we found that for this nucleobase, which is 2,6-diaminopurine, so now when you look at this, you might think, okay, these are the same pictures, but actually on the left-hand side, you have adenine as the purine base, on top of that TT dimer, the cyclobutane pyrimidine dimer, and on the right-hand side, you have 2,6-diaminopurine, so that's the difference. So that ring has one more amino group compared to this one. And this substantially changes the photochemical and photophysical properties. So the 2,6-diaminopurine trinucleotide absorbs at slightly lower energies, so longer wavelengths, but also the charge transfer states that are responsible for electron transfer, they lie at much lower energies than in the case of the biological canonical DNA fragment. And that essentially allows for efficient electron transfer. And when you look now at the blue curve, which is the potential energy surface for the TT dimer repair, associated with the electron transfer state, you can see that this is really an energetically favored reaction. And you can see what you can see here on the left-hand side is that this reaction leads to the breaking of the cyclobutane ring in one place. Whereas when you look at the biological one, it turns out that this reaction is not so favored energetically, and actually the direct photorelaxation of adenine is really preferred, whereas 2,6-diaminopirin doesn't undergo so efficient photorelaxation. It prefers to transfer the electron and repair the lesion. So that's what we found. We proved it experimentally, and since I'm running out of time, I won't tell much. It's just really to say thanks to Magda Zdravowicz, who did those experiments. And she found that for our trimers, including 2,6-diaminopirin, we get up to 92% of self-repair when we irradiate the damaged material. So this is remarkable because that's very, very photostable DNA oligomer. And when we actually look at the canonical ones, they undergo photo damage in 80%. So they can be repaired really in very small quantities. And I'll just quickly move to the end. So yes, we think 2,6-diaminopirin is prebiotically relevant. It is just slightly less photostable than adenine, but it can be formed prebiotically, and it selectively pairs with uracil, just like adenine does. And I'll just move to the end. So we are pushing these studies further. So we are starting new projects. We look at some proto-metabolic reaction networks. We look at prebiotic activation chemistry. We are starting to investigate non-enzymatic RNA self-replication from the point of view of molecular dynamics and QMMM, so quantum chemical calculations, and we're also developing machine learning potentials for that. And for these projects, I actually have, and for the one that I just showed to you several slides before, I actually have 3 postdoctoral positions available in my group. So please get in touch with me. One of the adverts is already available. And I'd like to thank my fantastic group for doing the work which I showed to you. Part of that I did myself, still part of that I did. Yeah, and just to very quickly wrap up, is just to tell you that UV light could have been an important selection factor and driver of prebiotic reactions. And when you think about the photostability of nucleic acids, this can be actually something very surprising, or you can find many surprising things, like just changing one small element, a nucleobase to a nucleobase, which can be perhaps slightly less photostable, can result in collective properties that make the whole DNA fragment much more photostable. And this is what we see in the case of DNA. So that can be counterintuitive in many senses, but photochemistry is not additive. Yeah, and what we think in the end is that there were probably really many reasons why nucleobases were selected. I mean, this very narrow range of 5 basic nucleobases. So photostability is probably just one of the elements, or maybe really an epiphenomenon, not the reason, but still it's intriguing to see this in biomolecular building blocks. So thank you very much for your attention, and I'd love to hear and answer your questions.